The U.S. CDC reports an average of 250,000 central line-associated bloodstream infections, or CLABSIs, occur annually in the United States across all hospital units. These infections are associated with 5 major sources of transmission, one of which is the needleless access site, which includes the needleless connector, the injection port, and the access ports. This critical access point accounts for approximately 12% of all CLABSIs. The Prevantics Device Swab is designed to help target this high-risk area. Unique to the market, the Prevantics Device Swab contains PDI's proprietary 3.15% chlorhexidine gluconate and 70% isopropyl alcohol formula, and is the only CHG product specifically tested and cleared by the FDA to disinfect needleless access sites prior to use. The Prevantics Device Swab has an intuitive prep pad format that is designed to easily disinfect grooves and crevices found on needleless access sites. Many clinicians refer to this practice as scrubbing and consider it an essential component of CLABSI prevention. In 3 simple steps, your facility can reduce the risk of contamination of needleless access sites, which include needleless connectors, injection ports, and access ports. Step 1. To get started, simply tear open the Prevantics Device Swab packet and remove the swab. Step 2. Apply swab to the needleless access site of the device by holding it between your thumb and index finger and scrub using repeated back and forth strokes for 5 seconds. Step 3. Allow the needleless access site to air dry for 5 seconds prior to entering the line or device. Do not blot or wipe dry. When compared to alcohol alone in a randomized crossover clinical trial, a CHG scrub used for a short time to disinfect needleless connectors before access resulted in significantly less contamination. The study's purpose was to determine the best method of disinfecting needleless connectors before accessing a central venous catheter in order to prevent the introduction of microbes to the bloodstream. The study compared 3.15% chlorhexidine gluconate, 70% isopropyl alcohol versus 70% isopropyl alcohol alone, using a 5-second versus a 15-second scrub for routine disinfection of needleless connectors on central venous catheters in an adult medical intensive care unit. Upon analysis of the data, it was determined that using the CHG scrub resulted in less needleless connector contamination when compared to alcohol alone. The difference was statistically significant for the shorter scrub time. Alcohol-impregnated caps are used by some facilities to disinfect needleless access sites after the clinician's last access. Nurses report they value the visual indication that a device has been disinfected and the physical barrier caps provide. While these are important components, caps are often not practical for disinfecting in between accesses, which is a critical practice in CLABSI prevention. Caps typically need to be applied for over 1 minute in order to disinfect. The Prevantics Device Swab only requires a 5-second scrub and 5-second dry time, making it practical for disinfecting between accesses. Additionally, the swab format of the Prevantics Device Swab easily conforms to surface shapes, bends, and tight grooves, such as the connection threads. This friction is important to actively remove bacteria. Prevantics Device Swab can be used for both terminal disinfection as well as disinfection between each access, while the caps can only be practically used for terminal disinfection. Another option is a handheld single-use device containing IPA. It contains IPA-saturated foam within a plastic casing. It looks like a cap, but it does not remain on the needleless access site after scrubbing. This device has 6 steps within its direction for use, with a minimum of a 10-second scrub and 5-second dry time. The Prevantics Device Swab, on the other hand, has an intuitive 3-step application process with only a 5-second scrub, 5-second dry time. The IPA device is comprised of hard plastic and IPA saturated foam. The cost of this product is typically double the cost of the Prevantics Device Swab. For many facilities, this product is too expensive to incorporate into protocols, especially for disinfecting after every access. The simple prep pad design of the Prevantics Device Swab allows for lower pricing. Additionally, Prevantics offers a 3.15% chlorhexidine gluconate and 70% isopropyl alcohol formula, whereas the IPA device is only available with an alcohol solution. The easy application process of the Prevantics Device Swab, combined with its CHG formula, provides better coverage than scrubs with alcohol alone. Independent studies have shown that the use of the Prevantics Device Swab is clinically proven to reduce CLABSIs if used as part of a bundle of evidence-based practices.